You guys can hear me, right?
Yes.
Okay, cool. So we have phototrophs versus chemotrophs. What's a phototroph?
Organisms that get their energy from light.
Right, exactly. So a phototroph is some type of organism that is doing photosynthesis to get energy from light, versus a chemotroph, which would be getting energy from chemicals of some type. And then we had our two types of chemotrophs, the chemolithotrophs and the chemoorganotrophs. What's a chemolithotroph?
Organism able to oxidize inorganic compounds?
Right. So it's an organism that's getting its energy from inorganic compounds, which would be any compound that is not based on carbon. In general, that means they're oxidizing those compounds to take electrons from them, and then those electrons are carrying the energy that they're getting out of the compounds. Then what's a chemoorganotroph?
Getting energy from organic compound and carbon?
Right. Right. So if a chemolithotroph is getting its energy from an inorganic compound, then a chemoorganotroph is getting its energy from an organic compound. So stripping electrons from it, oxidizing it, and taking the energy out in those electrons. And it so happens that usually for a chemoorganotroph, they would also be getting their carbon from the same source. Well, you know, like virtually always. So a chemoorganotroph is technically defined as getting energy out of organic compounds, but they would also generally get carbon from those compounds as well. Then you have autotrophs versus heterotrophs. What's an autotroph?
Gets carbon from inorganic molecule.
Right. So an organism that is getting its carbon from some inorganic molecule. So any molecule or any compound that is not based on carbon. And then a heterotroph?
From organic molecule.
Right. That's an organism getting its carbon from an organic source. So chemoorganotrophs would usually be heterotrophs as well. And we would usually combine those words to chemoorganoheterotroph. Cool. Then we have photosynthesis. So first, what's the difference between anoxygenic photosynthesis and oxygenic photosynthesis?
Anoxygenic is phototrophic bacteria that doesn't produce oxygen.
Right. So anoxygenic photosynthesis is just photosynthesis that does not produce oxygen. Out of all the organisms out there that can do photosynthesis, the only ones that do anoxygenic photosynthesis are bacteria. And then what's oxygenic photosynthesis?
Photosynthesis that produces oxygen.
Right, photosynthesis that does produce oxygen. So you have some bacteria that can do oxygenic photosynthesis, and then you have eukaryotes like algae and plants that do oxygenic photosynthesis as well. Then we had some pigments in photosynthesis. What's chlorophyll, or what is the function of chlorophyll in photosynthesis?
To absorb the red blue light so it looks green.
Well, to absorb the light, and it just so happens that since it absorbs red and blue light, it ends up looking green. Right, so chlorophyll's job is to absorb photons or absorb light so that the organisms can actually harvest the light energy. So if you didn't have a pigment to absorb the light, you wouldn't be able to do photosynthesis. The light's going to either just kind of pass through you or bounce right off of you. You need a pigment that can actually absorb the light. So chlorophyll is the main pigment absorbing light in certain photosynthetic organisms. What type of microbes would use chlorophyll to use or to absorb light for photosynthesis?
Eukaryotes.
Eukaryotes, yeah. And one more type.
The cyanobacteria.
Right. So chlorophyll is going to be used by plants, but of course plants aren't microbes. So your microbial eukaryotes, which would be mostly just algae, those are going to be using chlorophyll as their main pigment to absorb light for photosynthesis. And then out of the bacteria, only cyanobacteria will use chlorophyll. The rest of the bacteria that do photosynthesis use a different type of pigment. What pigment do the rest of bacteria use for photosynthesis?
Bacterial chlorophyll.
Right, bacteria chlorophyll. Cool. And then what do carotenoids do? Or carotenoids?
Protects the cells from damage.
Right. What type of damage?
The light.
Right. Right. So your carotenoids are protecting cells from light damage. Anytime the cell is in a high light level, you're going to have a lot of light coming in. Some of that light will be absorbed by the chlorophyll or the bacteria chlorophyll, but not all of it. And the rest of that light can end up actually harming the cell, for instance, by reacting with DNA or by making reactive oxygen species. So it's important for photosynthetic organisms to have the carotenoid pigments. Different carotenoids will absorb light at different wavelengths, so you can absorb like a kind of a whole wide range of light with those pigments, and then just all of the extra light that's coming in that doesn't get picked up by the chlorophyll or the bacteria chlorophyll gets picked up by the carotenoids instead. And that way the rest of the cell is protected from that light, and you do not have reactive oxygen species being generated by the light. So what type of photosynthetic organism would have carotenoid pigments? It's almost like a trick question, actually.
Is it all?
Yep, all of them, exactly. Every photosynthetic organism has carotenoid pigments. Otherwise, it'll be damaged by the light. Okay, cool. Then we have light reactions and dark reactions. So what are the light reactions?
That's where carbon is fixed.
That's going to be actually the other ones. But in the light reactions, you're getting something that you have to do the carbon fixation. So you do need to do the light reactions before you can fix any carbon.
Or photophosphorylation.
Yeah, photophosphorylation. What's basically happening in photophosphorylation?
Well, invaginations of the cell membrane in prokaryotes and chloroplast thylakoid membrane in the eukaryotes. It uses an electron donor and sunlight. It gives ATP and NADPH.
Right. So this would be occurring in some type of membrane. If we're talking about a eukaryote, it's happening in the thylakoid membrane of the chloroplast. If we're talking about a prokaryote, most of them will have, if they're photosynthetic, most of them will have little membrane invaginations, places where the cell membrane folds in and makes, little folds. So that's where your enzymes are for photophosphorylation. Photophosphorylation is going to require some type of electron donor, and it requires light, which means it's using pigments to absorb that light. And the result of photophosphorylation is going to be ATP and usually NADH or NADPH, which would be electron carriers. So then it's the ATP and the NADH or NADPH that you need for carbon fixation later on. So if you're not getting that ATP and the NADH or NADPH, you're not going to do carbon fixation. You have to have those things. So you're kind of transferring electrons from your electron donor in photophosphorylation to your NAD plus or your NADP plus, which would give you NADH or NADPH. And somehow you're also making ATP. So how is that process happening? How are you transferring those electrons, and why does that give you ATP?
From the Calvin cycle?
Let's see, so once you have your NADH or your NADPH and your ATP, you're going to use those things in the Calvin cycle to fix your carbon. But how are you going to get the electron onto the NADH or the NADPH in the first place?
From light and water?
If you're doing oxygenic photosynthesis, then yeah, you're going to be using light and water. What role is the water serving there?
The water gives oxygen.
You're going to take something off of the water, and then it turns into oxygen. What are you taking off of the water?
Hydrogen.
Hydrogen. You're going to take off a hydrogen ion for sure. And what else are you going to take off? What does a hydrogen atom consist of?
One proton
One proton and one more thing.
and one electron.
An atom. Sorry?
One electron.
One electron, yeah, exactly. So a hydrogen atom is a proton and electron. A hydrogen ion is just the proton. So if you have water, H2O, and you're going to transform that to oxygen, you need to take the hydrogen off of it. If one of the things you're taking off is just a hydrogen ion, just a proton, what's the other thing that you're taking off?
The electron.
The electron, exactly. Yeah, so in oxygenic photosynthesis, in the photophosphorylation, you are taking hydrogen ions off of the water, and you're also taking electrons off of the water, and that gives you oxygen. Well, you would technically need to do that to 2 waters, and then you can combine the oxygens from 2 waters to give you molecular oxygen, O2. Then those electrons are going to go where? Something that we also have. Sorry?
They're gonna go to ATP and ADP.
Ultimately, those electrons are going to go to the NADH or NADPH. They're going to get added to NAD plus or NADP plus, giving you NADH or NADPH. And while they're kind of moving, like on that journey from water to NADH or NADPH, you're going to do something with them that lets you then make ATP. What's kind of the pathway that they're moving through called? It's something that we also had in oxidative phosphorylation.
Electron transport.
Calvin cycle?
The electron transport chain, yeah. And then once they get done and they go to NADH or NADPH, that's going on to the Calvin cycle, which means that technically those electrons are moving on to the Calvin cycle. So, right, you have water at one end of your electron transport chain for photophosphorylation. You are stripping electrons off of it, stripping hydrogen ions off of it. That gives you just oxygen left over. Those electrons enter the electron transport chain. They hop from protein complex to protein complex along the chain till they get to the end, where they're going to be added to NAD+, giving you NADH, or they could be added to NADP+, giving you NADPH. Going back to the beginning of this electron transport chain, when you're actually going to strip those electrons off the water, you need something to do that. That's not like a normal thing that would happen to water. Water's usually pretty stable. So what are you using to strip electrons off of the water?
Is it the light?
Exactly. Yeah, the light. Right, so you have pigments. Part of the electron transport chain at the very beginning is these pigments that you're going to take. Well, they're gonna they're gonna absorb these photons of the light, and those photons have a lot of energy. Then they're going to transfer that energy to the water, and then the water has so much energy in it that it can't hold on to its electrons anymore. Those electrons are going to become really energized, and they will leave the water. Then you have the hydrogen ions leaving the water also, and you end up with just oxygen instead of water. Then those electrons are on the electron transport chain. So if you don't have the light there coming in, then there's no way to take the electrons off of the water. Water is just too stable a molecule to lose electrons unless you put a lot of energy into it. which would come from all that light that you're absorbing with your pigments. So for our oxygenic photosynthesis, we have, like, at the very beginning, a photosystem, as it's called, which is where you have all these pigments that are absorbing the light, transferring that energy to water, stripping electrons off of water. But if we want to get real technical, about it, though, actually the electrons are initially coming off of one of those pigments. They're coming off of a chlorophyll. Because if you're doing oxygenic photosynthesis, that means you're using chlorophyll. You're either a cyanobacteria or you're a eukaryote, and you use chlorophyll for your pigment. So light's getting absorbed by all these chlorophyll molecules in this photosystem, and one specific chlorophyll at the middle, or at the center of the photosystem, is going to receive all of the photons that the photosystem absorbs. And it's going to have its electrons stripped off of it. Then it's going to still have a lot of energy left over, and then it's going to strip electrons off of water to replace the ones that it lost. That gives you oxygen left over. Then those electrons are going to be traveling on the electron transport chain, hopping from complex to complex. When they're pretty close to the end of the chain, you have another special protein complex, which is just another photosystem that has pigments. So what would happen at that second photosystem?
Is it like the extra light goes through?
Yep, exactly. So that's your, well, I said it's the second photosystem because it's the one that comes second in the sequence, but we call it photosystem 1 because we found it first. So in this photosystem 1, the second photosystem, you have more pigments that absorb more photons, and that energy gets transferred to the electrons that are traveling on the electron transport chain. Those electrons get boosted up in their energy really high. And then shortly after that is when they get transferred to NAD+ or NADP+, giving you either NADH or NADPH. And then that would be moving on to the Calvin cycle. And then we still have one more thing which we need to account for, which is ATP. We're also getting ATP out of this. So how do you make ATP in photophosphorylation?
Also from the light?
It's also coming from the light, yeah. So your light energy is being transferred to these electrons traveling on the electron transport chain. So these electrons have a lot of extra energy on them. Even more so after they go through the second photosystem, photosystem 1, and then get boosted up in energy again. So as those electrons are traveling with all their extra energy that came from the light, what are they going to do that can help you make ATP?
They oxidize the electrons from water?
Let's see, I guess these would be the electrons that were oxidized from water, since they're the electrons that were stripped off of water.
Oh.
think when during the transfer, ATP synthase helps it cross the membrane.
Sorry?
They work with like ATP synthase.
Yeah, we're going to use
Hydrogen
ATP
ion.
synthase. Yeah, and hydrogen ions, exactly. So these electrons are going to do something with the hydrogen ions, or I should say the energy from the electrons is going to be used to do something with the hydrogen ions. What happens to those hydrogen ions?
They're pushed across the membrane.
Right, we're pumping them across the membrane, exactly. So your proteins of the electron transport chain are going to use some of that energy from the electrons that they're carrying to pump protons, to pump hydrogen ions across the cell membrane. If we're talking about a prokaryote, then they're going to be pumping it across the cell membrane. If we're talking about a eukaryote, they would be pumping it across the thylakoid membrane. And then that gives you a hydrogen ion gradient, a hydrogen ion concentration gradient. And then what does ATP synthase do with that gradient?
It will allow the hydrogen ion to cross the membrane.
Exactly. It's allowing those hydrogen ions to cross the membrane, going back the other way, down their concentration gradient. So they would be going from outside the cell to inside the cell if we're talking about a prokaryote. If we're talking about a eukaryote, they would be going backwards across the thylakoid membrane. In the process, ATP synthase is going to use the energy of those hydrogen ions flowing down their concentration gradient to make ATP by adding that free phosphate group onto ADP, giving you the ATP. So that part is pretty similar to oxidative phosphorylation, where you have electrons moving on an electron transport chain. You're using the energy of their motion to pump hydrogen ions across the membrane, and then ATP synthase lets those hydrogen ions flow back the other way and uses the energy to make ATP. What's different from oxidative phosphorylation is the fact that you are using light to strip electrons off of something that would not normally give up electrons, and then you're adding those electrons to an electron carrier. In oxidative phosphorylation, it's kind of backwards. You would be taking the electrons off of an electron carrier and then adding them to something that is really good at accepting electrons, like oxygen. And then let's see, one last thing I'll mention, or have you guys tell me that is. So since we're talking about prokaryotes a lot, anytime we're talking about a hydrogen ion concentration gradient for prokaryotes, that has a special name. What's the special name for a hydrogen ion concentration gradient across the cell membrane of a prokaryote?
The proton motive force?
Exactly. Yeah, the proton motive force. Cool. Okay, so that's oxygenic non-cyclic photophosphorylation, which would be the type that's used by like cyanobacteria or by eukaryotes. It's called non-cyclic because we're taking our electrons from one thing and then putting them on a different thing ultimately, which would be the electron carrier, the NADH or NADPH. So they don't just go directly back to where they started. Then we also have anoxygenic cyclic photophosphorylation, in which you are not producing oxygen and the electrons actually do end up back where they started. So how does anoxygenic cyclic photophosphorylation work? I guess where would the electrons be coming from? What's the electron donor for that type of photophosphorylation?
Mm.
So in our oxygenic photosynthesis, we said that at the beginning you have your photosystem. Your first photosystem technically is called photosystem 2 because we found it second. You actually don't need to know that. Just our first photosystem. And we're going to have pigments there absorbing light, funneling all that energy to the central chlorophyll pigment. And that chlorophyll, once it gets all this light energy, is going to have its electrons stripped off of it. And then to replace its electrons, it's going to take them off of water. So in our anoxygenic photophosphorylation, it's pretty similar, but we're not using chlorophyll anymore. We're using the other pigment, bacteria chlorophyll, because we're not doing oxygenic photosynthesis in cyanobacteria, basically. And we're not going to be making oxygen, which means we're not taking electrons off of water. But we do have a photocomplex that's absorbing light, that's going to funnel all of that light energy to a central bacteria chlorophyll. And then what's going to happen next?
The energy strips the electrons from the bacterial chlorophyll.
Right, exactly. So all this photocomplex has all these different pigments in it, absorbing all this light, all these photons, and all that energy ends up funneled to the, well, technically two central bacteria chlorophyll pigments. And once those pigments receive all of that energy, their electrons become so energized that they can't hang on to them anymore, and those electrons are stripped off of the pigments. So you have your central bacteria chlorophylls that are going to lose their electrons once they receive all of this light energy that's collected by the photocomplex. Then what happens to those electrons next?
They transfer it to the molecules.
Yeah, so you're going to be transferring those electrons to the molecules or the proteins and protein complexes of the electron transport chain. So the electrons are going to hop around on the electron transport chain. Where do they go ultimately?
These ones go back where they started, the central bacterial chlorophyll.
Exactly, yeah. So those electrons are ending up right back where they started on that central bacteria chlorophyll. By this point, when they get back to that bacterial chlorophyll pigment, that pigment doesn't have all this light energy anymore. It's not all energized, which means that now it's able to accept those electrons back. In the next cycle, it's going to absorb more light energy, become energized again. Whenever it's energized, it cannot hang on to its electrons. The electrons will leave, they enter the electron transport chain and hop around in a circle. By the time they get back to the central bacterial chlorophyll, it does not have all of that extra energy anymore, and it can accept the electrons back, and you're ready for another cycle again. So that's how the electron transport chain is operating in your anoxygenic cyclic photosynthesis. You are also still going to be pumping your hydrogen ions across the membrane and using that to make a proton motive force that ATP synthase is going to use to make ATP. And I guess the final thing is just how you're getting your NADH or your NADPH out of your cyclic photophosphorylation. And basically what's happening there is just that a few of these electrons, as they're moving around on the electron transport chain, are going to end up being diverted away from the electron transport chain and added to NAD+, giving you NADH, or NADP+, giving you NADP. Okay, so that's going to cover the light reactions for us. Then we have the dark reactions, which is the carbon fixation. And we had a number of different pathways for that, but the only one that you need to know at all is just the Calvin cycle. So what are some things that are required by the Calvin cycle?
CO2?
Right, you need CO2, exactly. That is your inorganic carbon source. Then the other things you need, you would be getting them from the light reactions.
Energy?
Energy, yeah, definitely. Where does that energy come from? Or I should say, what format is that energy in?
Will it be ATP?
Yep, yeah, exactly. So your energy in the form ATP, you need that for the Calvin cycle. You also need carbon dioxide, and then there's one last thing you need that also comes from the light.
NADP?
NADPH or NADH. Yeah, exactly. So you need electrons. Those electrons are going to come either from NADH or NADPH. So those are the kind of the three things you need. You need carbon dioxide, your source of carbon. You need ATP, your source of energy, and you need an electron carrier, either NADH or NADPH, which is your source of electrons. The ATP and the NADH or NADPH are both coming from the light reactions, so you have to do the light reactions first before you could do the Calvin cycle and the dark reactions. What are you going to make through the Calvin cycle?
Sugar?
Sugar, yeah, exactly. You're going to make some type of 6-carbon sugar. Generally, you'd be making glucose. Could also be other things. What are you going to use that sugar for?
It's used to build structures or for respiration.
Right. So you could either use that in cellular respiration to make more ATP if you need more ATP, or if you need to build like macromolecules and structures for the cell, you could use the glucose for that instead. Just depends on what you need more of right now. So for the Calvin cycle going in, you have carbon dioxide, ATP, and an electron carrier that you're using as an electron donor to donate those electrons to carbon dioxide, and then that gives you out on the other side glucose. And then let's see, which types of microbes will be using the Calvin cycle as opposed to some other cycle?
Yeah. Cyanobacteria?
Cyanobacteria, yeah, exactly. Who else?
Eukaryotes?
Eukaryotes. And who else?
The purple bacteria, but I don't know what a purple bacteria is.
Purple bacteria are bacteria that usually, well, they don't always look purple, but a lot of the time they look purple because they have carotenoid pigments. So those are photosynthetic bacteria that are using anoxygenic photosynthesis. And you have kind of the two types. You have purple sulfur bacteria and purple non-sulfur bacteria, just the ones that use sulfur and the ones that don't use sulfur. Yeah, so there's just a type of photosynthetic bacteria that is kind of classified based on the way it looks because of the pigments that it has. They're not even like all related to each other actually. Like a lot of them are related, but they're not all related. They just all have those pigments that give them frequently a purple color. So we have the cyanobacteria using the Calvin cycle. Those are doing oxygenic photosynthesis. We have the purple bacteria using the Calvin cycle. Those are doing anoxygenic photosynthesis. Then we have eukaryotes, specifically the photosynthetic eukaryotes are using the Calvin cycle. And then we also have one other type of microbe that can use the Calvin cycle.
Prokaryotes?
They are prokaryotes. Yeah, it's a type of prokaryote. Well, I guess it's like, it's like a functional classification, I guess. Prokaryotes with a specific type of metabolism that do not do photosynthesis, but do this other type of metabolism. So they're not phototrophs, but they're something else.
The chemolithotrophs?
Yep, chemolithotrophs, yeah. And what are the chemolithotrophs doing to get energy? Or where do they get energy from?
Inorganic compound.
Right, inorganic compounds. Cool. So you have phototrophs that use the Calvin cycle. You also have chemolithotrophs that use the Calvin cycle. For your phototrophs, the ones that use the Calvin cycle would be the eukaryotes, the cyanobacteria, and the purple bacteria. And then for the chemolithotrophs, we're not going to worry about which specific ones use the Calvin cycle. Just a lot of them do. Cool. And then the last thing about the Calvin cycle is Rubisco. What is Rubisco?
Is Ribulous biophosphate carbylase?
Yep, Ribulous phosphate carboxylase. So that long name, you do not need to know that long name for the test. What you would need to know is the function of the Rubisco for the test. So what is the Ribulous bisphosphate carboxylase doing?
It's a complex series of reactions in which starting chemical is generated.
That's going to be the Calvin cycle as a whole. And Rubisco is the
CO2. Yes. Add CO2 to the cycle?
Right, yeah, exactly. Yeah, cool. So your Calvin cycle is this long sequence of reactions where you end up where you started. The starting chemical is RuBP, or ribulose bisphosphate. But again, you don't need to know the long name, just RuBP is fine. Then you need to add carbon dioxide to that RuBP, and you're going to go through a complicated set of reactions where you're going to end up getting that carbon dioxide, well, not the carbon dioxide, but the carbon from it, you're going to end up getting that out as a product and ending up again RuBP, and then you can, fix other carbon. So first step is to add carbon dioxide to the RuBP, and Rubisco is the enzyme that catalyzes that step. So Rubisco is responsible for adding carbon dioxide to RuBP in the first step of the Calvin cycle. Because of that, we sometimes think of it as the most important enzyme because it's the first one involved in the Calvin cycle, which is used to fix carbon by so many different microbes and also by plants. And that fixed carbon is the source of carbon for chemotrophs like us that exist. So without the Rubisco, not only would plants be screwed, we'd be screwed too. Okay, so that's our Calvin cycle. Oh wait, one last thing to underline is the connection between the light reactions of photophosphorylation and the dark reactions of the Calvin cycle or whatever other pathway you're using. So how do the light reactions connect to the dark reactions?
I guess they connect in a way that the dark reactions won't occur until they get some And they get something from the light reaction.
Exactly, exactly. The dark reactions need stuff from the light reactions or else they cannot happen. What's the stuff that the dark reactions need from the light reactions?
Like the electrons?
Electrons is one of the things, yeah, definitely. And one more thing too.
Is it ATP?
ATP, exactly. It needs energy in the form of ATP. So the dark reactions have to get ATP. That ATP is coming from the light reactions. They also have to get electrons. Those electrons are coming from the light reactions in the form of NADH or NADPH, usually. Cool. And that's photosynthesis. So now we can go to nitrogen fixation. What is nitrogen fixation?
Reducing nitrate to ammonia?
That's going to be another process, actually. Let's see. So if you're reducing ammonia, you're not really going to reduce pneumonia or ammonia because it's already reduced, but you can oxidize it. Nitrogen fixation does involve ammonia, though. You're going to be reducing something to make ammonia in nitrogen fixation.
Then nitrate?
Nitrite.
Is it pyruvate?
Sorry? Pyruvate?
Is it
No.
no.
Sorry?
Yeah, I thought it was pyruvate.
No, it's not going to be pyruvate.
Is it nitrate? Or nitrogen?
It's nitrogen. Well, what type of, well, yeah. It's nitrogen as in molecular nitrogen in two. Yeah. So, I guess there's a few different processes that all involve nitrogen. So you want to make sure that you keep them straight because like they all are like, they're involving ammonia and nitrite and nitrate, and they all have a name that has something to do with nitrate. And nitrogen fixation, you're going from nitrogen, molecular nitrogen, into to ammonia. Then you also have nitrification, where you're going from ammonia first to nitrite, and then from nitrite to nitrate. Then you have denitrification, where you're going from nitrate or from nitrite to molecular nitrogen or nitrous oxide or nitric oxide. So in nitrogen fixation, you're going from molecular nitrogen, which is not organic, to ammonia, which actually is not an organic compound because it doesn't have carbon in it, but it is a compound that that organisms can use, that they can very easily incorporate into organic compounds they're making that do have nitrogen in the form of oh boy, amines. There we go. Right. So for this part of the test, or for this part, I guess for this chapter, all you really need to know is just what nitrogen fixation is there. That is just converting molecular nitrogen to ammonia, which is actually a reduction. So you're adding, in order to get molecular nitrogen to be ammonia, you have to add electrons to it and reduce it, and then ammonia is like the reduced form. Then for the next chapter, chapter 15, is that for true? Oh no, never mind. I'm sorry, you guys. Yeah, no, you do need to know the other nitrogen processes for this chapter as well. They're just not at this point on the study guide. But actually, maybe we should talk about them together since they're like so close to each other. So nitrogen fixation is reducing molecular nitrogen to ammonia. Then nitrification is called that because we're taking ammonia and we're producing nitrite or nitrate. Denitrification is called that because you're taking nitrogen in the form of either nitrate or nitrite and you're converting it to a gas, either molecular nitrogen or nitrous oxide or nitric oxide. All those three things are gases. So when you do that, it's going to leave. The gas will go up into the atmosphere. So you're losing nitrogen from wherever this is occurring, the water or the soil. Nitrogen fixation is called that because you're taking a form that is not useful, molecular nitrogen, and putting it into a form that is useful, ammonia, to a lot of organisms. So you're fixing it for them. So I guess that's the rationale behind those names, which is probably what you want to use to remember like which is which there. I guess mostly the nitrogen fixation name makes sense and the denitrification name makes sense since you're removing nitrogen from something for denitrification and since you're putting nitrogen into a usable format for nitrogen fixation. And then the other processes must be nitrification. Then for nitrification, there's actually two different processes inside of that. Both of them are oxidations. So you have ammonia oxidation and then you have nitrite oxidation. What are you doing in ammonia oxidation?
Ammonia, it's oxidize to nitrate?
See, yes. Well, wait. Ultimately, in nitrification, you're going to be oxidizing ammonia to nitrate. But first, you actually have to oxidize it to nitrite. So in the whole process of nitrification, you're oxidizing ammonia to nitrate. In just ammonia oxidation, you're oxidizing ammonia to nitrite. And then in the next step, nitrite oxidation, you're oxidizing nitrite to nitrate. I think what I should do is put like, I should make some type of summary for all this nitrogen stuff. Yeah, so I will make that and I will post that. Right, so in ammonia oxidation, part of nitrification, you're oxidizing ammonia to nitrite, and then in nitrite oxidation, which is the second part, you're oxidizing nitrite to nitrate. Then if you remember that in nitrification, the two parts of it are ammonia oxidation and nitrite oxidation, and that first you're doing ammonia oxidation, then you can know that you must be oxidizing ammonia to the thing that is needed for the next process, which is nitrite. And then the nitrite is oxidizing that, or nitrite oxidation is oxidizing that nitrite to the final product, which is your nitrate. Okay. And then under nitrification, well, related to nitrification, I should say, we also have another process, which is anammox, which is another type of ammonia oxidation, which is different from nitrification, because in nitrification, ammonia is oxidized to nitrite and then to nitrate. In anammox, ammonia is oxidized to molecular nitrogen. So that means that anammox is basically the reverse process of nitrogen fixation. In nitrogen fixation, you reduce molecular nitrogen to ammonia. In anammox, you oxidize ammonia to molecular nitrogen. Okay. So that covers those parts of the study guide. And I will make like a summary sheet, I guess, that just shows those reactions and lists out which one is which. Okay. So looping back up, we have assimilative reduction and dissimilative reduction. What is assimilative reduction?
Assimilative is where nitrate, I think sulfate, and carbon dioxide can be reduced to incorporate into organic compounds?
Right. So for nitrate, for sulfate, or for carbon dioxide, you can reduce those things for multiple purposes. You be reducing them in order to make an organic compound, or you could be reducing them to make energy. If you're reducing them to make organic compounds or cellular structures, that is assimilative reduction. If you're reducing them for energy, that's the other one, that's dissimilative reduction. So if you're assimilating something, then you're kind of taking it into yourself and incorporating it into yourself. So if you're reducing the nitrate, sulfate, or carbon dioxide assimilatively, you're adding it into organic molecules for the cell, incorporating it into the structures of the cell. Then let's see. Which one is more common? Is assimilative reduction more common or dissimilative reduction more common?
I think assimilative is common.
Yeah, assimilative is more common. Every organism needs like amino groups for their amino acids and their nucleotides. Every organism needs carbon for their organic molecules, and every organism needs sulfur in the form of sulfhydryl groups for a couple of the amino acids that have sulfur in them. So, like, definitely not every organism can do that with carbon dioxide, because if you're doing that, that would be an autotroph. But every organism is going to be able to do like at least one of those assimilative reductions, either incorporating nitrate or sulfate or carbon dioxide or some combination of them. Organisms that are doing those things for energy are much less common. Cool. And then let's see. Yeah, actually, if you remember that stuff, then you should be good. So just the assimilative reduction is reducing those things to make them into cellular structures. Dissimilative reduction is doing it for energy, and assimilative reduction is more common. Then that's all you need to know about that. Okay, then we have our common electron donors. So we've got a list of donors there. Just memorize the list. That's it. And then you would need to know, out of the molecules on that list, which ones would be used as an electron donor by a chemolithotroph. So there, you're just looking for the ones that are not organic compounds. The ones that are organic compounds would be used by chemoorganotrophs. The ones that are not organic compounds are the ones used by chemolithotrophs. So for the chemolithotrophs, which electron donors out of that list would they be able to use?
Hydrogen?
Hydrogen, yeah, exactly. What else?
Sulfur?
Sulfur, definitely. And you have kind of two forms of sulfur on there, just plain sulfur and hydrogen sulfide, or H2S. Both of them are for chemolithotrophs.
Iron?
Iron, definitely inorganic. So that one's for chemolithotrophs too. And then you have two more.
Sulfite?
Sulfite. Yeah, the hydrogen sulfide, that's definitely going to be for chemolithotrophs. Sulfite would also be used by chemolithotrophs. But that one you're not going to need to know because it's not on the list.
So, nitrite?
Nitrite, yeah, definitely. That one doesn't have carbon in it, so it is not organic. And one more.
Ammonia?
Ammonia, yeah, exactly. Right, so your molecular hydrogen, your hydrogen sulfide, the H2S, the sulfur, the iron, the ammonia, and the nitrite. All those things are inorganic. They do not have carbon in them, which lets you know they're inorganic for sure. So if you're using those things as electron donors for energy, that means you are a chemolithotroph. Hydrocarbons are organic molecules. Methanol and methane, or CH4, those are all organic molecules. So if you're using those things as electron donors to get energy, that makes you a chemoorganotroph. So I'm going to have kind of questions just asking you, like, which of these is an electron donor? And so you would want to recognize, like, the ones, it would be either the ones on this list here for the common electron donors or the ones in the list below for the common electron acceptors. So just memorize those two lists. And then for the electron donors, I'll also ask you which ones would be used by chemolithotrophs. You're just going to look for the ones that are inorganic compounds. And then finally, I'll also have some questions asking you which of those are going to be oxidized and which ones are reduced. And there you just know that if it's an electron donor, that means that, which is it? Is it going to be oxidized or reduced?
Reduce.
Well, you're going to be reducing something for sure. But if we're donating an electron from this compound, like whoever's getting that electron is going to be reduced, but this compound itself would be oxidized.
Oxidized, yeah.
Hence, oil rig. Oxidation is losing, reduction is gaining. So if you have hydrogen sulfide and that's an electron donor for you, that means you're taking electrons off of it, so you're oxidizing that compound. So all of your electron donors, those ones are oxidized. All of your electron acceptors, those ones are reduced. So if I asked you, like, which of these compounds is oxidized in respiration or in one carbon metabolism, and I give you a list that has some of those donors on it and some of those acceptors, you would be looking for the ones that are donors. If I'm asking you which ones are reduced, now you're going to be looking for the acceptors. Does that make sense?
Yes.
Cool. Okay, so now we have nitrification. I'm going to make you guys a summary sheet for that. And moving on to the one carbon metabolism. What is one carbon metabolism?
Using compounds with just one carbon for energy.
Right. Using a compound that only has one carbon in it for energy. Some of those could be organic compounds, some of them might be inorganic. So if you're using carbon dioxide, that's inorganic. If you're using methane, it's organic. So we have the acetogens versus the methanogens. What's the difference between acetogens and methanogens?
Acetogens, they generate acetate from carbon dioxide, and the methanogens, they generate methane from carbon dioxide.
Yeah, exactly. Yeah. So acetogens making acetate from carbon dioxide, methanogens making methane from carbon dioxide. The ogen ending there tells you that it's generating something. So acetogen must be generating acetate, the methanogen must be generating methane, and both of them are actually making those things from carbon dioxide. Then the other types of one-carbon metabolizers that we're talking about are the methylotrophs, and then the subset of methylotrophs, which is the methanotrophs. So what's a methylotroph?
Is it the main area where the colony is?
Sorry?
It says it's microbes using organic compounds without the C-to-C bond for energy.
Yeah, exactly. So a methylotroph is some type of microbe that is using organic compounds that don't have carbon-carbon bonds for energy. If you are on an organic compound without carbon-carbon bonds, there's only one way to be that type of there's only one way to be that type of compound, which is to not have multiple carbons. Organic compounds that have multiple carbons always have those carbons attached to each other, which means they always have carbon-carbon bonds. So only organic compounds that just have one carbon would be able to not have carbon-carbon bonds. There's a variety of organic compounds that just have one carbon, but the two main ones, like by far the most common ones, are going to be methane and methanol. So a methylotroph usually is going to be using either methane or methanol as an energy source and as a carbon source, technically. Those are both organic compounds that do not have carbon-carbon bonds. So within the methylotrophs, we have a subset of organisms called the methanotrophs. What do the methanotrophs use for energy?
Methane?
Methane, yeah. So the name is kind of telling you. Methanotroph is using methane for energy. And there, if you just know those definitions, you'll be good. I guess it could be easy probably to mix up methanogen with methanotroph, since they both have methane in the name. But if you remember that the gen there in methanogen means that it's generating methane, then you'll know the methanogen makes methane from carbon dioxide. The methanotroph uses methane for energy. Right, and I guess when I say that the methanotroph uses methane for energy, the methylotroph is using either methanol or methane for energy. That means that the methane or the methanol there are serving as electron donors. So they're oxidizing those compounds to get energy from their electrons. Cool. And then the last thing there is just the lactic acid bacteria. So what are lactic acid bacteria?
The bacteria used for the fermentation process.
Right, they're doing fermentation. What type of fermentation do they do?
The lactic acid.
Right, lactic acid fermentation. So, as that's fairly straightforward, lactic acid bacteria do lactic acid fermentation. So in order to get energy, they are doing fermentation, the specific type of fermentation that produces lactic acid. Cool. So that's chapter 14. Then we just have a little bit on chapter 15. Mostly about more about phototrophs. So out of the phototrophs, which ones are able to fix nitrogen?
Isotrophs?
Sorry?
Diisotrophs?
Oh, diazotrophs. Yeah. So actually, I didn't put the word diazotroph on the test, but technically any microbe that is fixing nitrogen is counting as a diazotroph. So all the phototrophs that fix nitrogen are indeed diazotrophs, for sure. And then as far as like different groups of phototrophs, which ones are able to do nitrogen fixation?
Is it like the purple non-sulfur bacteria?
Yes, purple non-sulfur bacteria, those ones fix nitrogen for sure. Who else? There's just two more that fix nitrogen.
Cyanobacteria?
Cyanobacteria? Yes, definitely. And one more group.
The heliobacteria?
Yep, heliobacteria, exactly. So out of our bacterial phototrophs, we have three groups that fix nitrogen: the heliobacteria, the cyanobacteria, and the purple the purple non-sulfur bacteria. And then which ones are heterotrophs out of the phototrophs? Which ones are heterotrophs as well?
The heliobacteria?
Heliobacteria again? Yes.
The purple non-sulfur?
The purple, the purple non-sulfur, yeah.
Green non-sulfur bacteria?
Yes, the green non-sulfur bacteria also, yeah. And one more big group.
The aerobic and oxygenic phototrophs?
Yeah, the aerobic and oxygenic phototrophs. So technically, we also have the acetobacteria and the gematomonadites that are photoheterotrophs, but technically, those are not, like, we only know one species out of each of those groups that are phototrophs at all. So I just didn't put them on the test. So we have those 4 groups: the heliobacteria, the purple non-sulfur bacteria, the green non-sulfur bacteria, and the aerobic and oxygenic phototrophs. Those are all heterotrophs. Cool. So then we have nitrogen fixation in cyanobacteria. So how do cyanobacteria fix nitrogen?
They use the enzyme nitrogenase.
Yeah, exactly. They're going to use nitrogenase. Technically, everybody who's fixing nitrogen is using nitrogenase. What inhibits nitrogenase?
Oxygen.
Oxygen, exactly. So oxygen will irreversibly inhibit nitrogenase, making it completely unusable. So the cyanobacteria have to have some type of a way to separate their nitrogen fixation from their oxygenic photosynthesis that creates oxygen. What are some ways that cyanobacteria keep their nitrogenase away from oxygen?
By doing photosynthesis at night?
Right. Some of them will do, well, actually the other way around. Some of them will do nitrogen fixation at night and photosynthesis in the day. So during the day, they're basically not making nitrogenase. They don't have any nitrogenase, and they are, doing their photosynthesis, making oxygen. At nighttime, you technically can't do photosynthesis anyways because there's not light. So at that point, they're going to start making their nitrogenase enzyme and doing the nitrogen fixation. What's another way that cyanobacteria could keep oxygen away from their nitrogenase?
By becoming heterocytes?
Right. So some of them will form like a special type of cell called the heterocyst, which is able to do nitrogen fixation, and it basically doesn't do photosynthesis, and it's going to not let oxygen in. So the heterocyst is going to have really thick walls, so oxygen cannot cross the walls, and it's just doing nitrogen fixation in there, not doing the oxygenic photosynthesis. And then what do heterocysts do with the neighboring cells?
They trade the nitrogen.
Right, they're trading fixed nitrogen with the cells around them. Exactly. So the cells around them might give them something that they got from doing oxygenic photosynthesis. The heterocysts, which don't do oxygenic photosynthesis, would be sending out fixed nitrogen and getting back maybe like electron carriers. Cool. And cyanobacteria are super important, and there's a few reasons why. So what are some things that are really important about cyanobacteria historically?
The Great Oxidation Event.
The Great Oxidation Event, exactly, definitely. That's one of the major ones. So cyanobacteria are the reason why we have oxygen in our atmosphere. And then one more really important thing about cyanobacteria in history. evolutionary history.
Well, I guess that's the same. I was gonna say the first organisms to synthesize oxygen, but I guess that's the reason we have O2.
Right, that's the Great Oxidation Event. It's something related to eukaryotes, to phototrophic eukaryotes.
They're usually blue-green due to chlorophyll.
So that's not it, but that is a clue to it, because the cyanobacteria have the chlorophyll pigment. Other bacteria that are doing photosynthesis don't have chlorophyll. They would have bacteria chlorophyll instead. And then when you look at eukaryotes, they also have chlorophyll, like the cyanobacteria. So why is it that both the eukaryotes and the cyanobacteria have chlorophyll? Why eukaryotes have bacteria chlorophyll?
Because they have the phycobilin pigments.
It's not going to be the phycobilins. Some of them will have phycobilins, actually, for sure, but some of them don't. But all of them will have the chlorophyll. How did eukaryotes get their chlorophyll? Something from a previous chapter.
Because they absorb light.
Well, that's why they need the chlorophyll for sure, to absorb light for them. But how did they get chlorophyll evolutionarily? How did eukaryotes get photosynthesis evolutionarily?
They fix carbon and nitrogen.
That is something that cyanobacteria do, most of them. And I guess most eukaryotes that do photosynthesis would not be fixing nitrogen, actually. They would be relying on bacteria for that. Well, I should say most non-microbial eukaryotes would be relying on microbes for nitrogen fixation. Right, so photosynthesis evolved in bacteria. It never evolved normally in eukaryotes. How did eukaryotes get photosynthesis?
Is it the different types of photosynthesis?
It's kind of related to that. So you have, in bacteria, you have oxygenic photosynthesis with chlorophyll in the cyanobacteria. Other types of bacteria that do photosynthesis do anoxygenic photosynthesis and use bacteria chlorophyll. Eukaryotes all do it like cyanobacteria. They all have chlorophyll and they all do oxygenic photosynthesis. Why are they all doing photosynthesis in the same way as cyanobacteria?
Because they have oxygen as a waste product from water?
Let's see, it's not going to be because of that. That is, I guess, the result of the photosynthesis that they're doing. Since it's oxygenic, they are making oxygen as a waste product from water. But why are they doing oxygenic photosynthesis and not anoxygenic photosynthesis? How did they get photosynthesis? So yeah, I think we went over this in chapter 2. Yeah, we talked about it in chapter 2. I guess what organelle would a eukaryote use for photosynthesis?
Um, chloroplast.
Chloroplast. Yeah, exactly. So every eukaryote that is doing photosynthesis has the chloroplast organelle that they're using to do it. Where do chloroplasts come from?
You said where do they come from?
Yeah.
The chloroplasts develop independently, right?
Right, they divide independently from the rest of the cell. So it's not like a normal organelle at all. But it is like the mitochondria, which also divides independently from the rest of the cell. And then the chloroplasts and mitochondria have their own DNA.
And the symbiotic theory.
Yes, exactly, into symbiosis. So cyanobacteria are important because they cause the great oxidation event and are the source of oxygen in our atmosphere. They're also important because they are the source of the chloroplast. A cyanobacterium was absorbed by a eukaryote many ages ago and turned into a chloroplast. That's the reason why eukaryotes have photosynthesis. So all eukaryotes that have photosynthesis do it the same way that cyanobacteria do it because they got it from cyanobacteria through endosymbiosis. So those are the two reasons why cyanobacteria are so important. Okay. Let's see, then we have the green sulfur bacteria, which mostly, I just wanted to look at the chlorosomes, or, mostly on the test is just the chlorosomes. What is a chlorosome?
structures full of pigments that absorb light?
Yes, right. So the chlorosome is like a membrane fold, like folded membranes inside the cell of a green sulfur bacteria. And it's got a bunch of pigments in it, like a bunch of pigments, and they're absorbing light, like a lot of light. And then they're going to be funneling that light into the photocomplex for photosynthesis. So any phototrophic organism is going to be having like a lot of different pigments, and they're absorbing light at all these different chlorophylls or bacteria chlorophylls or even phycobilins, and funneling that light into the center of some photocomplex or photosystem. But the chlorosome is special just basically because it's like real big and has so many pigments, more pigments than what's normal. So it's absorbing an incredible amount of light and funneling it all into the photocomplex for photosynthesis. So because these green sulfur bacteria have chlorosomes, where are they able to live that another phototroph could not live?
Lakes and oceans?
There's a lot that live in lakes and oceans, but it's where they are in the lakes and the oceans that's unusual.
Deep in water?
Deep, yeah,
At the bottom
exactly.
of the ocean, like where there's
Right.
no light, oxygen?
Right. Yeah. So the green sulfur bacteria can live really deep in the water. They just need like the barest, the teensiest amount of light. Even at the very bottom of the ocean, in a few places where you have those deep sea vents, it's just, there's not like light per se, but it's very hot, and then that heat is heating the rocks, and then the rocks start glowing a little bit. Not that you wouldn't be able to see them glow because it's like infrared radiation, but if you had like night vision goggles, then you could see it. So the green sulfur bacteria are able to harvest that light with their chlorosomes and use it for photosynthesis. So they need the least light out of any phototroph because they use the chlorosomes to collect light really, really efficiently. So even if there's just the barest bit of light there, they're going to catch it. They're going to catch it all. They're going to soak it all up in the chlorosomes, and they can do photosynthesis with it, which lets them live deeper in the water than any other phototroph can, in places where there's less light than any other phototroph would be able to survive with, basically. Cool. So there's your green sulfur bacteria. Then we have the sulfate and sulfur reducers and who would be living with them. So if you are a sulfate or a sulfur reducer, what are you doing?
You're gaining electrons.
Right, you're putting electrons onto the sulfur or the sulfate, which means that like at the end of your electron transport chain, you're using the sulfur or the sulfate as your final electron acceptor, which would mean that you're doing anaerobic respiration of some type. Well, I shouldn't say of some type, of the sulfate or sulfur type. So that's what you're doing for energy. Once you have added electrons to the sulfur or the sulfate, what compound do you get out of that?
Oxygen?
So you're not going to get, well, oh, you know what? I don't know. I'm going to say no, you don't get oxygen, but I don't know what you get. Obviously, that oxygen is going to go somewhere. It's going to end up in some other type of molecule, but I don't know what molecule. I don't know where it goes. Where does the sulfur go?
It's used as an electron acceptor?
Your sulfur, if you're doing sulfur reduction, the sulfur is used as an electron acceptor. If you're doing sulfate reduction, the sulfate is used as an electron acceptor. Either way, you're going to be making the same compound ultimately, which is some compound that contains sulfur.
Oh,
And it also
hydrogen
contains, yeah,
sulfide?
yeah, the hydrogen sulfide, yeah. So when you add those electrons to the sulfur or the sulfate and reduce them, you end up with hydrogen sulfide. So, who would like to live with those sulfate or sulfur reducers? It's anybody who likes to use hydrogen sulfide. So what's an example of a type of bacteria that likes to use hydrogen sulfide for its metabolism?
Delta Proteobacteria and phylum Proteobacteria.
You do have some in the phylum Proteobacteria, definitely. You're not going to need to know that, though. You're not going to need to know what phylum they're in. Just what metabolism do they use?
Is it like the obligate anaerobes?
They're going to be, some of them could be aerobic, some of them could be anaerobic. Some of them are obligate aerobes. What would they be doing with the hydrogen sulfide, though? So let's see.
Respiration?
Yes, respiration. Well, I should say some of them are doing respiration. Some of them are actually doing photosynthesis. But in any case, like whichever one they're doing, they're going to be oxidizing that hydrogen sulfide, right? To make it in the first place, you're going to reduce something, either sulfur or sulfate. So then to use the hydrogen sulfide, since it's already reduced, you're going to oxidize it using it as an electron donor. So some of them would be using it as an electron donor for respiration. Some of them would be using it as an electron donor for photosynthesis. What's a group of bacteria that would use hydrogen sulfide as an electron donor for respiration?
The chemolithotrophs?
They are chemolithotrophs, yeah. And we can get even more specific than that.
Is it Criniarchaeota?
Too specific. Yeah, some of them are in phylum Crinarchaeota. But that's not on the test. So we'll just call them sulfur oxidizers. So sulfur oxidizers use hydrogen sulfide as an electron donor for respiration. They're chemolithotrophs. They like to live with sulfur reducers that produce their hydrogen sulfide for them. And then we also have some phototrophs that use hydrogen sulfide. It's actually the purple sulfur bacteria and the green sulfur bacteria. Both of them are using hydrogen sulfide as an electron donor for their own photosynthesis. So, who likes to live with the sulfate and sulfur reducers? It's going to be the sulfur oxidizers, the purple sulfur bacteria, and the green sulfur bacteria. The sulfur oxidizers like to live with the sulfur and sulfate reducers because they're able to use hydrogen sulfide that's made by those sulfate and sulfur reducers as an electron donor for respiration. The purple and green sulfur bacteria like to live with sulfate and sulfur reducers because they can use hydrogen sulfide as an electron donor for photosynthesis. Cool. And we're like super close to the end of chapter 15, so I think we'll just finish it up. The next point on there, which processes conflict with oxygen, they're just listed there, just sulfur oxidation, nitrogen fixation, iron oxidation. All those processes use an enzyme that is inhibited by, well, nitrogen fixation uses an enzyme that's inhibited by oxygen, and then sulfur oxidation and iron oxidation, if you have oxygen around, the oxygen will just go ahead and oxidize the sulfur or the iron instead of the bacteria doing it. So if you just remember those three processes, you'll be good on that. And then the ecological importance of denitrification, nitrogen fixation, and methanotrophy. So what's the importance of denitrification in terms of its impact on the environment?
For denitrification, is in agricultural soil they drain NH3 from fertilizers and convert to NO2 as a greenhouse gas.
Right. So they can produce a variety of gases from ammonia for their metabolism, one of them being N2O. N2O is nitrous oxide, and it's actually greenhouse gas. So that's bad for like farmers because they need to use more fertilizer, but it's also bad for the environment because now you're putting a greenhouse gas into the environment. Then what's the importance of nitrogen fixation?
Is it because it reduces molecular nitrogen to ammonia, and ammonia is used for fertilizers?
It is because it reduces molecular nitrogen to ammonia. Ammonia is used for fertilizer. So it's related. It's related to It's related to that. It's related to ammonia and fertilizer. So why would we put ammonia in fertilizer? Why is it beneficial?
I think because it can be broken down to the nitrite and nitrate, and the legumes will be able to use the nitrate. I'm just making this up. I don't know.
You're fine. So you have some plants that are like that want it in the form of like nitrate, like the legumes. Right, so those guys are definitely going to be taking ammonia. Well, they won't be doing it themselves, but they'll have bacteria living with them who are taking that ammonia, converting it to nitrate for them through nitrification, and then they're able to use that. The average plant, though, is actually able to use ammonia directly as a source of nitrogen, and it's just that it's a source of nitrogen, right? So the air is mostly nitrogen, molecular nitrogen. Your plants are like, living in the air. There's nitrogen all around them, but it's molecular nitrogen, and they can't use that. So if you want them to grow good, you need to make sure that they have enough nitrogen, and they are able to use nitrogen usually in the form of ammonia, except for the ones that use it in the form of nitrate. So most plants are using nitrogen in the form of ammonia, and that's why ammonia is in fertilizer. Just making sure you have nitrogen in a format that plants can use. So these nitrogen-fixing bacteria are taking molecular nitrogen that plants cannot use and putting it into the form of ammonia, which most plants can use. For the ones that can't use ammonia, then you would also need to have the nitrifiers come, convert that ammonia to nitrate so they can use that nitrate. So nitrogen fixation is important just because it's providing other organisms with a source of nitrogen that is in a format that they can use, ammonia. And then methanotrophy. What is methanotrophy?
Using methane?
Yep. Using methane for energy. So why is that ecologically important? Or why is that important for the environment?
Because it's a very strong greenhouse gas.
Right. Methane's a really strong greenhouse gas. And methanotrophs use it, so then that's getting rid of that greenhouse gas, which is, a good thing. So for methanotrophy, methanotrophs are bringing down the levels of methane, using up that methane, which helps us, get rid of some of our greenhouse gases so we have less climate change. For the denitrifiers, on the other hand, they're producing nitrous oxide, which is a greenhouse gas, so that's actually causing climate change. And then also, we didn't mention before, but from the previous chapter, denitrifiers also are producing nitric oxide, which falls back down to the earth in the form of acid rain. So when you have more denitrifiers, you're going to get more greenhouse gases, more climate change, and more acid rain. When you have more methanotrophs, you're going to have less greenhouse gases. And then when you have more nitrogen fixers, you just have more ammonia that's available for other organisms to use. Okay, so that's that. Cool. Did anybody have any questions they wanted to ask about this stuff? All right, for sure. So in our next review session, we'll go over chapter 16, then we'll loop back around and hit chapter 14 again probably, or anything you guys want to hit. And I'm going to make I don't know exactly what it's going to be yet, you guys. Something some type of summary showing like all these different nitrogen processes, like labeled and like next to each other, so it's easier to kind of keep them straight as far as which is which. All right, cool. So thank you for coming, you guys. I hope it was helpful. And I hope you guys have a good night.
Thank you too.
Thanks. Bye.
Thank you.